Welcome, everyone. Today we're going to talk about something that has quietly become one of the most critical risks in modern drug development: nitrosamines, especially in emerging drug classes like peptides, oligonucleotides, and novel modalities. For years, nitrosamines were considered a small molecule problem, but that assumption no longer holds. In this video, I'll walk you through where these risks come from, why they're often missed, and how teams can practically manage them before they turn into late-stage surprises. Let me start with a story that many development teams will recognize. A peptide program looks clean. The synthesis is well controlled, the impurity profile is stable, and everyone assumes nitrosamines aren't relevant. Then, late in development, sometimes during stability or regulatory review, trace nitrosamines are detected. Suddenly, timelines stretch, investigations begin, and confidence drops. The key lesson here is simple but uncomfortable. Nitrosamines are no longer just a small molecule problem. They're showing up in places we didn't expect, and often when it's most expensive to fix them. Traditionally, nitrosamine risk was easy to explain. Amines react with nitrosating agents. End of story. But emerging modalities add layers of complexity. Peptides introduce protecting groups, coupling reagents, and scavengers. Oligonucleotides rely on phosphoramidite chemistry, solid supports, and specialized buffers. Novel modalities combine synthetic chemistry with biological systems, advanced excipients, and hybrid formulations. Each layer introduces new precursors, new impurities, and new degradation pathways. That's why regulators now expect nitrosamine risk evaluations across all drug modalities, not just tablets and capsules. Peptides are especially misleading. They feel safe because they're amino acid based, but the chemistry used to make them tells a different story. Many peptide syntheses rely on secondary amines, especially piperidine during FMOC deprotection. If nitrites are present from solvents, water, or reagents, nitrosamine formation becomes chemically possible. There's also a second risk window: storage. Peptides can degrade over time, generating small amine fragments that form nitrosamines long after manufacturing is complete. The practical takeaway? Peptide developers must monitor both process-related and storage-related nitrosamine risks, especially for injectables. Oligonucleotides bring a completely different challenge set. Their synthesis uses phosphoramidite chemistry, which involves tertiary amines that can act as nitrosatable species. Solid phase supports may carry residual amines, and quenching agents can introduce additional reactive impurities. Then comes formulation. Buffers can contain trace nitrites, and oligonucleotides are often designed for long-term stability, increasing the window for slow nitrosamine formation. Unlike tablets, you can't simply extract and test these products. Success requires deep understanding of oligonucleotide chemistry and tailored analytical strategies. Novel modalities push complexity even further. Antibody-drug conjugates introduce nitrosamine risk during payload and linker synthesis. Lipid nanoparticles involve complex lipid interactions and degradation pathways. mRNA formulations rely heavily on buffers and long-term stability, both of which can contribute to nitrosamine formation. Here, nitrosamines don't form through a single step. They emerge across synthesis, formulation, storage, and degradation. That's why, for novel modalities, risk assessment is just as important as analytical testing. Traditional nitrosamine testing wasn't designed for these systems. Peptides and oligonucleotides suppress ionization, making trace detection harder. Lipid systems interfere with extraction, leading to poor recovery. Biological matrices hide nitrosamines behind complex backgrounds. That's why modern labs rely on LCH-RMS, isotope-labeled standards, and matrix-matched validation. And here's an important regulatory insight. Sensitivity alone isn't enough. Regulators expect selectivity, robustness, and scientifically defensible methods. Across successful programs, a few best practices consistently work. First, start with a chemistry-based risk assessment, not a checklist. Identify nitrosatable amines and nitrosating agents early. Second, optimize sample preparation to avoid degradation during extraction. Third, use high-resolution mass spectrometry with isotope-labeled standards to reduce false positives. And finally, test stability samples. Many nitrosamines form slowly, not during synthesis, but over time. These strategies have been validated across peptides, oligonucleotides, and novel modalities. Regulatory expectations are now very clear. If your drug contains amines, complex chemistry, or a long shelf life, nitrosamine risk must be addressed. That means a documented risk assessment, justified testing strategies, and data showing long-term control. Agencies understand complexity, but they expect proactive science, not reactive fixes. And importantly, these expectations apply to all drug modalities, not just traditional APIs. Across the industry, the same lesson keeps repeating. Late discovery costs more. Teams that integrate nitrosamine testing early avoid reformulation, delays, and regulatory uncertainty. Looking ahead, testing strategies must become smarter, more chemistry-driven, and integrated earlier in development. Understanding your chemistry remains the strongest control strategy for predicting and preventing nitrosamine formation. Thank you for watching. If you found this helpful, follow the channel for more insights into pharmaceutical analytics and regulatory science.